Okay, we are starting with the next session, which is going to be chaired by Dr. Uzair Chaudhary from Ontario Institute of Agrologists, Canada. Professor Dr. Uzair Chaudhary. Professor Dr. Uzair Chaudhary is a President, National Alliance for Safe Food Management Director, Global Halal Services. He is a founder and president of National Alliance for Safe Food Management, not only in the Global Halal worker, but also a doctoral in the Dodhabai Institute of Higher Education, Karachi. He has many professional affiliations, including Chief Organizer, Food Technologies Community, Karachi. Next, we are going to request Dr. Mohammad Awais Khan to please co-chair the session. Dr. Mohammad Awais Khan. He's not here, okay. Next, we will also request Dr. Kashifa, please, if you can co-chair the session. Dr. Kashifa Nagma. Dr. Kashifa Nagma in her MPhil and a double gold medalist holder in her PhD work from GC University. Her research interests are in fisheries and aquaculture. Thank you so much, ma'am and sir. The first presentation is going to be by Professor Dr. Uzair Chaudhary from Ontario Institute of Agrologists, Canada. He is going to be telling us something about trends in weed science, research and management in 2050, that is 2050. Please welcome Dr. Uzair Chaudhary.
Honorable guests, colleagues, inertia from academia and research institutions from Pakistan and abroad, the organizers of this conference, ladies and gentlemen. The presentation has in the title, Trends in Weed Science Research and Management in 2050. So what will be the scenario? This presentation is basically focused on a bit crisis caused because of the pesticides. What is the status globally? Because we heard a lot of presentation this morning and there was a concern shown while we saw the presentation on cancers particularly. Let me start with the concept. I knew I am going to an institution where there will be many students as well. So a few of my slides are very basic definitions and I will skip them quickly. What are the weeds? Weeds are basically undesired plants. They are the organisms out of place, and a newer definition in area of agronomy, crop production is volunteer crops, plants emerging in subsequent crop season. So a crop plant can become a weed. So something which is precious at one time become a weed at another time. Why weed is a menace? Weed plants are also good when they are medicinal, right? We may extract some medicinal valuable chemicals out of them. But they become a problem when they are out of place, when they come and compete with our crops and take away all nutrients and water, which is a very precious commodity these days. There was a recent study published in 2018. Weed Science Society of America assigned a task to the scientists in Kansas State University, the place where I graduated my master and a PhD in weed sciences. And they had a 7 years average study done to review the status, how far these weeds are competing and taking away the capital of investment in the crops. Only on two crops, corn and soybean, they observed about $43 billion worth loss because of the wheat competition. Look at the situation in Ontario. The wheat crop, if you look at the graph, from 10% to maximum 53% wheat infestation, is resultant for the yield losses. What are the weed challenges? Smaller the organisms, more the population. You must have seen and heard, elephants have fewer kids than the mice. So same is the weeds. The tremendous seed bank which they leave into a field area, that is one problem. Most weeds see dormancy breaks when crop grows. So they come in competition. When the crop has a favorable weather to grow, the weeds also enjoy the same weather to grow. And they avail the farm tillage advantage. When we till the soil, what we do is we unearth the soil and bring the seeds up. Seeds are getting all aeration to have their respiration gone up. So they have the sunlight availability and they can grow faster and compete with the crop plant. Speedy growth habit wins competition for water, nutrients, light and space. So I'll show you the pictures in the next slides. We will see how they have win the space for the crop with the crop. They are very resilient. So they induce tolerance and resistance against biotic and abiotic stresses. So most part of my presentation here will be on this resilient characteristic of the weed, because today there is a lot of resistance induced into the weeds and they have become a havoc to control in the areas of the world where they have tremendously used herbicides. What is herbicide resistance? Before I jump over to herbicide resistance caused, let's know ability of plant to grow after exposure to a lethal dose of herbicide. Globally, 350 plus weed species and biotypes have been reported develop resistance against different herbicides. This table will just cover few of the herbicides which are known weeds have developed resistance. And this table shows the 2,4-D is a very common growth regulator type herbicide which proliferates cell growth and it clogs the transportation of solutes and nutrients in the plant, and that results a cause of death of the plant. Phenoxy herbicides we call them, the group of phenoxy herbicides. Since 1945, we look at 1963, so in the beginning there is a large gap when they discovered the resistance. Nobody knew that these plants will develop a resistance. So in the beginning of the era when the herbicides were introduced, nobody was knowing and nobody pointed it out. Look at the bottom part with the glyphosate, it's also called Roundup. Within 3 years, the resistance were reported. Why? Because of the awareness, because of the information system, so knowledge proliferations. So people were more aware about the health because of the side effects of the herbicides. And by the way, the herbicides are the most, in terms of tonnage or kilograms used over the all list of pesticides. So insecticides are the second after herbicides. These are the few of the weeds which are reported have a lot of resistance to the glyphosate. Glyphosate is Roundup ready crop, they say. So nowadays there are crops developed by Monsanto. They have genes in them which are resistant to the herbicide glyphosate. Glyphosate is a broad spectrum herbicide. It will kill all plants, including the weeds and crops. So they have put a gene which can resist glyphosate in the crop plant. So the crop plant is safe, but the weeds will die. So these are the few weeds which have developed resistance against glyphosate like the crop. This is a situation in Ontario. One of our colleague friends, Dr. Sikama, he had a survey and he got these pictures of the weeds which has developed resistance to glyphosate. A little close up with the corn, you can see how competition of the weed is with the crop. They have taken away all space and they are also having the competition for the nutrients which were provided for the crop. Look at the situation on the map of the US. All these states, these are the few weeds. I won't spend time naming them all, but this gives you a scenario how the map of the North America, they have developed resistance against the glyphosate Roundup. A number of resistant species, as I mentioned already, there are different modes of action. There are about 8 modes of actions against which are applied, and I won't go more details on what are those enzymes and how these are resisted. But look at the map, it will give you an idea that about 160 species are ALS inhibitors, and there are photosynthetic inhibitor herbicide, this blue line. So this is second, about 100. So almost about 400 plus weeds have been resistant to these mode of actions. As far as the scenario of the all developing countries, because most herbicides are used in US, Monsanto is having a huge business in US. So people have been using glyphosate tremendously. So if you look at the status, how number of unique resistance has been seen in the US is the highest. And the second is the Canada, because Canada being neighbor with US, we try to follow up the US lines. Crop-wise situation, number of herbicide-resistant species, there are a lot more in wheat because wheat is a more staple crop. A lot more areas in Saskatchewan, Alberta, in prairies in Canada. We have all wheat dominant area. And look at the corn and soybean, 80%, 90% of the crop grown in the US is glyphosate resistant, and they're mainly basically corn and soybean. Now, when the resistance developed, there are a lot of concerns. North America, people are educated, media is very strong, as we have now the media coming up strong in Pakistan too. Look at the situation in North America where the media has more legal framework and autonomy. About 75% people are now having a concern why glyphosate is here. Monsanto itself is a big giant multinational, billion-dollar business. They are resisting. Recently in California, there was a sue against Monsanto, and the guy who was who used to spray glyphosate into the field areas in the school. So he got lymphoma, cancer. So he won the sue, and he was probably about $300 million. So there are about 2,000 people more waiting to come up against Roundup. Roundup has been used in North America about 35, 40 years now. When I went to study US, at that time glyphosate was introduced, 5 years old product, and it was a very potent product. So it has a lot of commercial value. What are the emerging challenges in 2050? First of all is global population. All of you are aware of we are going to be 9 billion. So total world land mass is 149 million kilometers square, and the scientists, they say arable land is about 12 to 18% out of it. And about 27 million kilometers is the total which is arable. And at the average, they say it is available 1 acre to feed a person. According to that, it should be not more than 5.5 billion people to be fed out of the existing resource. Few more challenges. Exacerbated climate change. We did hear some presentation on climate change. There is scant water resource, and for irrigation, for crop to grow, you need water. And we are hearing there is no water available for drinking of the human being. And you are all aware of the crisis likely Pakistan is going to confront. Nutrients, the fertilizers, we import a lot of fertilizers from abroad. Fertilizer is very costly ingredient. And there's not only one fertilizer, there's macro and micronutrients which our crops needs. So energy, to run your tractor, to run your machinery for the farming, you need investment. So that is also one challenge. Urbanization and land desertification. People are moving to the cities from the villages. And lands, they are unattended. They are getting desertifications, deforestation. Environmental pollution, contamination in food chain. I'll go a little quick. Unpredictable health budget changes. I was watching in US, the health budget has been increased from 6,000 to about 9,000 now per person every year. In Canada also, it is $450 billion total federal budget goes to the health. All health is covered there. So when there will be a lot of pollution and pesticide problems, so a lot more cases will call for health, and then there will be a lot more changes in the budgetary provisions. Continuum of weed resistance against limited mode of action of pesticides. This is also a challenge. Precision in current practices. We will need to have a very precise way of farming where we have to invest less on chemicals. What are the tentative solutions then? Reduce global population. How can we reduce global population? It has yet not been possible. I remember when I was a student in Pakistan, we were West Pakistan 4 crore and East Pakistan was 6 crore. Now West Pakistan is only 20 crore, right? So you can imagine population control and not in the very much control. Social values. Increase arable land, can we do that? There is a climate change concerns. It's lot more energy dependent. It's very costly, we can't do that. Of course, increase production efficiency. Still there is a room to increase production efficiency. The example of green revolution is in front of us. In 60s, I remember when we were in grade 12, grade 11 students, green revolution.
Thank you so very much. The next presentation is by Professor Dr. Mushtaq Ahmed, Director, Herbarium and QAU Botanic Gardens, Department of Plant Sciences, Quaid-e-Azam University, Islamabad. He is going to be talking about emerging research trends for sustainable utilization of plant diversity to industry in Pakistan. Please welcome.
Bismillahirrahmanirrahim. I am Dr. Mushtaq from Quaid-e-Azam University. I would like to discuss about the emerging trends in plant diversity leads toward the industry to the botany, from botany to the industry. I am from Quaid-e-Azam University, which is the top-ranking university in the country, and Pakistan with a unique biogeography, with a unique diversity of the plants for the industries. As far as the plant diversity provide us food, medicines and energy. So the three basic challenges is facing by the Pakistan as well as the world: food, energy and medicines. All these are fulfilled by the plant diversity, and the Pakistan has a rich diversity that are used for all these challenges. As far as my research group is working in Pakistan as well as in the world, our focal areas are the medicinal plant for health, non-edible oil plant for the energy, and food plants as a nutraceutical industry in Pakistan. You know that Pakistan is a diverse country. So there is a rich diversity of the wild edible fruits and wild vegetables. They are used as a food as well as a nutraceutical product by the nutraceutical industry in the country. How these nutraceutical plants leads toward the industries? We have identified about 255 plant species. They have a greater potential to use as a nutraceutical product for the nutrition, for the food, for public health in Pakistan. We compare the nutraceutical analysis, nutraceutical contents of these wild edible fruits and vegetables in comparison to the cultivated fruits and vegetables. There are basically three approaches. First approach is to identify the wild edible fruits and vegetables by comparing the specimens in the herbarium, in the botanical garden and in the field. Second step towards the drying and preservation of these nutraceutical products in the laboratory without using the chemicals is healthy for the food. The third step is towards the nutraceutical profilings to compare the minerals, nutrients present in these wild fruits and vegetables and also present in the cultivated plants. So this is the stage for the experimental work about the analysis of the nutraceutical products. The fourth step is towards the identification of the new active antioxidant from these wild fruits and vegetables. We have published one of the study recently, the potential of the wild edible fruits to combat a large number of the diseases as well as the food. So this paper is published in Journal of Ethnopharmacology from Pakistan to combat the health as well as the food crisis in Pakistan. Our focal areas about the number of diseases that are invaded in Pakistan, the diabetes, the hepatitis, the cancers and diabetes are the among of the top diseases in Pakistan that can be treated by using these nutraceutical products present in Pakistan. Our approach is bottom-up approach. We identify the plants, then screen these plants for the bioactive compounds, then these bioactive compounds leads toward the clinical analysis and then leads toward the new drug discovery development by using a lot of analytical techniques as well as the indigenous techniques. Basic approach is from bottom-up approach. Ethnopharmacology leads toward developmental approach, that is the biological and the phytochemical screening. Then this screening leads toward the natural drug discovery development by using the bioactive compound from the indigenous medicinal plant from Pakistan. I think I have a short of time, but I just give a glimpses. One of the drugs that we have identified, Pakistan, China, pioneer drugs, that is the patent drugs by China and Pakistan, is used for the epilepsy, first time introduced in this 2018. So you just see the paper published by one of the prestigious journals about the potential of these drugs from Pakistan to combat the epilepsy and the skeletal disorders by using these pioneer drugs. Another important drugs that we have identified from plants are the natural dietaries. Because the people, they have obesity, so the obesity leads to number of diseases. By using the natural drugs, the obesity is reduced, then these obesities then reduce the large number of diseases like the heart and blood pressure and cancer as well. We have to authenticate these herbal drugs by comparing a lot of techniques from basic identification towards the chemical profilings. Another important, the honey we use every day is directed by a number of other resources. So one of my projects is about the identification of the quality of the honey by studying the melissopaleontology, that is the pollen flora linked towards the honey. So these are just studies that are published in 2017 and 18 about the good products from the plants that is used towards the nutraceutical and the pharmaceutical industries. Just to see the publication, recently we have published, because short of time, I have just gave a glimpses about the potential of the nutraceutical pharmaceutical product from Pakistan. These are just approaches from field towards the industry. Another important plant we have identified that is Capparis residua, Dela Urdu mein isko kehte hain. That is one of the important plants that is used for the bone cancer. We have identified this species and to identify some active compounds that are used for the bone therapy as well. A book published just in last week Plant and Human Health, we have contributed this chapter about the public health and the potential of the medicinal plant for the public health of Pakistan. Another important source is the identification of the calcium constituents in the medicinal plants. We have published a paper about the calcium producing plants from Pakistan that are used for the bone therapy and the bone arthritis, rheumatisms. Another paper is published by the European Journal is about the musculoskeletal disorder by using the medicinal plant. A paper published on the management of the diabetes by such type of the nutraceutical products from the plants, just published in 2018. Another important area in Pakistan is the challenges for the herbal markets. So a study is published from Pakistan that we imported a lot of medicinal plants from neighboring countries like Iran, India and other Afghanistan. So we have great potential for these markets. Another important plant we have identified in 2018 about the oral disorder, that is the mouth cancer. So this is another important drug from Pakistan we have identified between Chengdu Institute of Biology in China and Pakistan Qaeda University. One of the important medicinal plants for skin cancer. So we have studied this plant between Pakistan and Chinese Academy of Sciences in Chengdu Institute of Biology. A paper published on bacteria drug from Pakistan and China by CID and QAU joint venture. These are the joint venture between Chengdu Institute of Biology China and Qaeda University about the natural drugs used for a number of cancer therapy. Another important plant, Contd, that is one of the plants used for the kidney dialysis, kidney cancer. So one of my PhD students did a work with University of California to identify some new active biocompounds for kidney disorder by using this indigenous plant from Pakistan. These are some glimpses of my student in US. Another important plant we have identified from Balochistan, that is Ferula Okoda. My recently produced Dr. Zain did a work on, he identified about 14 active compounds that are used for the cancer therapy on cell line experiment with the joint venture of Mississippi University and Qaeda University.
tin and nickel 1.5 millimolar. And you can see here order of resistance, the highly resistant is chromium. So you can see growth curve. Organism was grown in the presence of LP medium, minimal salt medium, and minimal salt medium containing chromium. Chromate reductase, but relative activity was determined. You can see with reference to pH, which was 8, and with reference to temperature, which was 40, and some metal ions were also added to see the activity of the organism. And you can see here just magnesium just has helped to increase the activity. Otherwise, other metal ions did not change the activity. These are some antioxidant parameters. You can see glutathione transferase, catalase, superoxidase mutase, peroxidase, and peroxidase. You can see peroxidase has been increased more profoundly as compared to the control. Metal processing ability was also checked, and you can see almost more than 60% chromium has been removed from the medium. Maybe this removal is of two types, maybe absorption and maybe adsorption. I mean, in a combination, organism has ability to remove metals from the environment more than 90%. But here you can say the provisions of ability, you can see 60, and other one is absorption is like more than 18% from the environment. Just to cooperate in the sense, assess, you can see the SEM as compared to control. The shape of the organism is a little bit altered in the presence of chromium to show chromium is not good for the proper growth. You can see here EDX, energy density X-ray. The aim is just to see, this one is treated. You can see here is chromium. I don't know you can see here or not. This one is chromium. Just to infer that organism has ability to uptake chromium. Of course, when organism will uptake the matter inside the cell, of course, the other enzymes will reduce the chromium 6 to chromium 3. Next is FTR, just to see the phosphate, sugar moiety, and other changes. And hopefully, maybe you understand this. The changes here, the changes indicate these phosphate or these carbohydrates molecules. Usually, the membrane surface is negative, and all of you are familiar that the matter is cation in this sense positive and negative. I mean, there are some interaction. It indicates that organism is doing some work present in the environment containing metal ions. I mean, these changes indicate that organism is interacting with metal. First, metal is adsorbed, and then after adsorption, it will uptaken by the organism. Next, you can see another parameter just to see. There are two systems. One is antioxidant molecule, which I have already shown, maybe in the form of GST and ascorbic peroxidase, catalase, and SOD. Maybe there are some molecules which are known as antioxidant molecules, maybe in the form of glutathione, GSH, or non-protein thiols. You can see the non-protein thiol and GS has been profoundly increased in the presence of metal to combat the negative effects of the chromium present in the environment. Next is, this is just a proposed diagram based on our result. You can see chromium enter inside the, I don't know, actually it is not working as I don't know. Chromium enters inside the cell, not specific process, just sulfate. This is chromate uptake here. Chromium is converted into chromium, different valencies, basically say 3 to 6. Here you can see intracellular chromate reductase converted 6 into eventually into chromium 3, RAS generation, here antioxidant molecule. Maybe one of the example is just GSH. GSH converted hydrogen peroxide, which is toxic for living organism or living system, but eventually converted into water, and everybody knows water is life or system. No life can exist without water. To combat this oxidative stress through antioxidant molecule or antioxidant enzymes. Here, combat oxidative stress through antioxidant enzymes. We mentioned just peroxidase because it has been increased profoundly in the presence of metal ions, mainly chromium, and you can see if extra chromium is available in the system, of course finally it will be effluxed out from the system.
Thank you so much. Next we have a Skype lecture by Professor Christine Lee-Matre, Sheffield University, UK. Professor Lee-Matre is an internationally renowned researcher in investigating musculoskeletal novel therapies. She is also a researcher in natural agent as a potential therapeutics for leukemia. She is currently the module leader for the biology of the diseases and intervertebral disc degenerations. She has 63 high index publications. Today she will be giving us a Skype lecture on treatment of leukemia with pomegranate juice. Please welcome the Skype lecture from Professor Christine.
And cell cycles can be analysed using flow cytometry and pyridium iodide stain. The pyridium iodide basically stains the DNA within the cells, and this allows you to identify cells which are in the G0 or G1 phase, where they have one copy of the DNA, those within the synthesis phase, where the DNA is being synthesised and therefore increasing levels of DNA will be seen, and within the mitosis or G2 phase, where you've now got double the amount of DNA. So if we look at the cell cycle results, next slide, what you can see within all of our leukemia cell lines shown by the top 8 panels, we see a rest of cells within the S phase following pomegranate juice. This effect was also seen within the control cells, the CD133 cells, suggesting that we do get some arrest of cell cycle within the normal cells, as we do within the leukemia ones, but we don't see the induction of apoptosis in those cells. Now you may be thinking, well, all this effect is just because we're putting juice in there, could it just be effects of the isotonic or osmolarity changes or pH changes? So if we do controls for this, looking at pH adjusted media and the percentage live cells, you can see it has very little effect on the induction of apoptosis, as opposed to the pomegranate juice treatment shown in the upper right corner, where you can see high induction of apoptosis and late phase dead cells. Likewise, isotonic or osmolarity changes also have no effect on cell viability, demonstrating there's something within the pomegranate juice that is producing the bioactive component. Sorry, I said next slide. So we're now up to the slide which has got the cell line and the source and whether they're more affected or least affected. Hopefully you can keep a look at the slides. So we demonstrated that when you look across a whole host of different cell lines, the lymphoid cell lines appear to be more sensitive to myeloid cell lines, and then hematopoietic stem cells, which are our normal control cells, were the least sensitive. So we then went on to investigate which agents within the pomegranate juice could be responsible for these properties. So we undertook solid phase extraction to separate out the compounds within pomegranate juice according to their chemical structure. Next slide. So when you put the pomegranate juice through the solid phase extraction column, there should be a little picture of a column with 5 tubes with different colours in the bottom now on the slide. And what you'll see when you actually do solid phase extraction, the phase which is coloured, which indicates where the polyphenols are, is the acetalnitrile phase. And when we take these phases and treat them with cells, indeed you see the bioactive components are within the acetalnitrile phase. So next slide, up to where it's caspase-free results for 48 hours. And on here you can see that the only effects are observed with the acetalnitrile phases, where you see a good induction of apoptosis within the cell lines. So then we went on to look at the total polyphenolic compounds within the different agents to see which ones have the phenolic compounds. So next slide. And as you can see, it is the acetalnitrile phase that contains the high levels of polyphenolic compounds. Next slide. So then if we look at the acetalnitrile fraction in more detail, with further analysis, so next slide, we can see with an X5 PI that you get good induction of apoptosis and cellular death with the treatment of the acetalnitrile fraction. Next slide. Again, we get induction of caspase 3 positivity as well, which is a marker of apoptosis. Next slide. So then we went on to do liquid chromatography and mass spectrometry to identify what the components within the different fractions were, particularly within the acetalnitrile fraction, to identify what these potential agents could be within the pomegranate juice, which were providing these potential anti-cancer actions. Next slide. So if we look at the results from the acetalnitrile fraction, you can see there's a number of polyphenolic compounds which have been identified, and these have been putatively identified as phenylcollagen, delphinidin, and cyanidin, plus a number of other agents. Next slide. So then we went on to investigate which of these phenolic agents could actually be the active agents which are producing the potential anti-cancerous properties. Next slide. So the pomegranate contains a whole host of different phenolic agents. So we should be up to the slide with a picture of pomegranate in the middle and then all of the different compounds around the outside. So if we go to the next slide, what we've then done is treated cells with some of the common agents which are found in pomegranates that have been suggested by other studies to demonstrate bioactive compounds such as EGCG, which is actually a key component of green tea. Now what you can see when you treat leukemic cells with EGCG is that some of the cell lines are quite sensitive, whereas THP1s, which are a myeloid cell line, show very little sensitivity. In contrast, on the next slide, when we treat with quercetin, you can see that all of the cell lines are responsive to the quercetin treatment. Next slide. But what we found is the most active agents were phenylcollagen, which showed good sensitivity within 3 of the cell lines, but less so in THP1s again. And next slide, delphinidin, which showed good responsive rates across all of the different cell lines. So we then went on the next slide to investigate what apoptosis pathways are being activated by these agents. So I've just got some examples of results, so I've not got time to show you them all. But if you treat the cells with delphinidin, then you get induction of caspase 8. And this is indicating that we're getting extrinsic pathways of apoptosis being induced. However, on the next slide, when you look at caspase 9, you also see induction of caspase 9. So therefore, we're getting activation of both the extrinsic, suggesting binding to the death receptors, and also intrinsic pathways, suggesting activation of cytochrome C and the mitochondrial response. Next slide. So in conclusion, bioactive agents within pomegranate juice have induced apoptosis and cell cycle arrest, preferentially in leukemia cell lines, which is really important if we want to develop new therapies which are going to target the leukemia cells in preference to the normal cells within the body, which current chemotherapy agents do not do. The major bioactive components of pomegranate were found to be delphinidin and phenylcollagen, and they were shown to induce apoptosis both for the intrinsic and extrinsic apoptosis. And we're now looking at the molecular pathways of these actions to find out whereabouts these agents are binding and how exactly they are having the actions. So I'd just like to acknowledge, in particular, the PhD student, Haytham Douali, who was a PhD student from Saudi Arabia who did this work a few years ago now, Dr. Nikki Jordan Mahi, my fellow collaborator, and Gordon McDougall from the Scottish Crop Institute for the help with the mass spectrometry analysis. I'll happily take some questions if there's a way we can do that. Thank you.
Bismillahirrahmanirrahim. Ladies and gentlemen, Assalamu alaikum. It's an honor for me to be standing here and present my work to all these educated people in front of me. Who doesn't want a healthy life? To look good, to feel good. We all do because it's natural. It's a human tendency to feel good. But to what extent are we ready to go for it? At what cost are we ready to look good? My topic of presentation today is perception of fad diet and weight loss tendencies among male and female young adults. I will start with a brief introduction, then I will discuss the objectives and methodology, results, and end this presentation with the conclusion. Fad diets. A fad diet is a weight loss diet that becomes very popular quickly and then fails out of favor just as quickly for various reasons. Some commonly known fad diets include controlled carbohydrates, high carbohydrate, low fat, liquid diets, diet pills, herbal diets. Now, what is the link between fad diets and weight loss tendencies? As the weight loss tendencies among young adults increase, the popularity of fad diets increases just as much. The cycle starts when you are motivated to lose weight. But we are so desperate to lose weight, we want it to be quick, we want a magical solution that we choose the wrong path, and we go towards fad dieting. And it does work for a while. We lose weight, we shed some fat, but then comes the plateau stage. No change, no weight loss, no weight gain. And that is followed by episodes of binge eating. You eat a lot. You restrict yourself and then you start eating like a mad person. And that results in the weight gain. When you weight gain, you do not like what you see. The results are very unlike. And that makes you unsatisfied of your body again. And then the cycle continues. You are motivated to lose weight again and you choose to go for fad dieting, and the cycle goes on. So the main objectives of this research were to assess the weight loss beliefs and fad diet concepts among young adults, to compare weight loss tendencies, and most importantly, check the effects of fad diets among male and female adults. The methodology followed was simple. It was a survey cross-sectional study. The process of simple random sampling was used. 80 females, 80 males were chosen from university, mostly were students. The instrument administered was self-constructed questionnaire, and it was a very carefully constructed questionnaire. The data analyzed was using SPSS version 20. Results. It was very interesting to know that when asked if the public was aware of the term fad diet or not, 54 males and 45 females said that they were not aware of the term fad diets. And yet, when given the options, we observed that the most popular of the fad diet was the controlled carbohydrates, cutting down all the simple sugars and carbohydrates from the diet. On second, it was food combining, eating certain foods with the others to improve their effect. And then there was the low carb diet, liquid diets, diet pills. 46% of the people said other, which probably included Atkins and keto diets, which are gaining popularity every day. This is the most interesting piece of data that I found personally very interesting. 33%, that was 61 students, had at some point in their lives followed fad diets. What was even more interesting was when asked for how long they had followed the fad diet, 37% could not follow it for more than a month, and 34% said that they couldn't follow the diet for more than a week. That shows how inconsistent the fad diets are. Results of the longest diet followed. 49% observed no change at all. That was because it is very difficult to carry on the fad diet. It is very difficult to restrict yourself for that long of a time period. 36% lost weight and gained it back, as we discussed before in the fad diet cycle, and only out of 100, 14% students said that they lost weight and kept it off. The most common side effect of fad diet was irritability, and then we had diarrhea, vomiting, and even heart problems. Recommending a fad diet.
When asked if they would recommend the fat diet to other people, 72% said no. And that was just as expected, because when you follow fat diet, you know what the evils of a fat diet are. Conclusion. In reaction to a prevailing trend of slenderness, more and more people are jumping on the wagon of fat diet bandwagon. The more people seem to diet, the more weight they seem to gain. That is because a recent survey of WHO says that obesity is increasing day by day, and yet the popularity of fat diet is increasing as well. That shows that fat diets are not helping in reducing the obesity or gain weight in the world. Most people today believe that to be slim, it is necessary to be healthy. To be slim is necessary to be healthy, and they are mistaken, because most diets are so unhealthy as to be useless in the long run. When dieting to lose weight, never ever lose the sight of health and balance, because that's all that you want at the end of the day. You want to be healthy and you want to be eating a balanced diet with all the essential nutrients. So always consult a health professional or dietitian before considering dieting. That is because we all have different body types. Our hormone level affect our body stature, and that's why there are multiple factors that have to be considered. Don't Google the diets, don't go for fat diets, don't just cut your carbs or increase the proteins. Consult a professional, because a diet should transform your body and not just change it in a superficial manner. You don't just want to shed the weight, you want to feel healthy. And always eat a balanced diet with plenty of variety within the calculated calories range. Thank you so much.
Dr. Faryad Akram from Home Economics Department, Lahore College for Women University, to give a presentation on studies on comparative antioxidant potential of raw and roasted nuts, pulses and pea. Please welcome Ms. Faryad Akram.
My name is Faryad Akram, and my topic of presentation is studies on comparative antioxidant potential of raw and roasted nuts, pulses and seeds. When we came to the diagram of nuts, pulses and seeds, we know that nuts, pulses and seeds represent an important part of our daily life, and is important and is important part of the human diet in many countries, which represent many health benefits. And also many epidemiological studies associated their consumption with many health benefits, and often consumed after roasting. That may destroy some bioactive compounds, but they also retain antioxidant potential, which helps in prevention of many degenerative diseases like cancer, like Alzheimer's disease. Research has shown that not getting enough magnesium may lead to hypertension, diabetes, heart disease and heart depression. All nuts and seeds gives a rich amount of magnesium, so by taking these in our diet, we can prevent these all diseases. In my studies, I took two seeds, 2 nuts and 2 pulses. The 2 seeds were sesame seeds and the sunflower seeds, and among nuts, pistachio and the walnut nuts was selected. Among pulses, soybean and small lentil were selected. So, in the part of the methodology in this study, I selected 2 nuts, pulses and seeds. This is the comparative study. The objective of this study is to analyze the antioxidant capacity of all these nuts, pulses and seeds, and also provide nutritional information of samples and recommend nut and seed separate with their nutrition facts. So that's my part of the methodology. First, I grinded the samples and also roasted all of these samples. After grinding, extraction of all samples was done, and the total antioxidant capacity of all samples is determined by the DPPA screening assay. So let's come to the results. Among pulses examined, the pulses was pulses was soybean and small lentil. The total antioxidant capacity of pulses was soybean had a higher antioxidant capacity than small lentil. And among walnuts and pistachio, walnut shows high antioxidant capacity than pistachio. And among seeds, sunflower seeds had a great antioxidant potential than sesame seeds. So this is the total comparison of all raw and roasted nuts, pulses and seeds. Roasting time was depend of 30 minutes. You can see that walnut. You can see that the black one is the walnut, which has a high antioxidant capacity, and the grey one is the roasted walnut. You can see that after roasting 30 minutes time, the roasted walnut decreases total antioxidant capacity. The fourth one is the pistachio. The total antioxidant capacity of pistachio is also higher than roasted pistachio. The blue one is the sunflower seeds and is also higher than total roasted antioxidant capacity. So same as the sesame seeds and roasted sesame seed, the roasted sesame seeds show low amount of total antioxidant capacity than raw sesame seeds. The green one is the soybean. Soybean show high capacity of antioxidant capacity, and the light green was the roasted soybean, which show decrease in the amount of total antioxidant capacity. The last one is the small lentil. Small lentil also show highest total antioxidant capacity as compared to the roasted small lentil. So we can conclude that the raw things which we consume in our daily life are rich in total antioxidant capacity than the roasted pulses, nuts and seeds. These are all the nutritional information of nuts, pulses and seeds which I have used in this study. Walnuts, pistachio, all amount of calories, fats, proteins, fibers are mentioned in that table. So the next one is recommendation. I highly recommend that recipe. So I can, that is very useful in our daily lives. We should use in our daily life. That is 1 cup of walnut half cup of sunflower kernels, half cup sesame seed, 1 teaspoon honey, 1 teaspoon water. All these combined in a food processor, stored spread in a refrigerator at approximately 1/3 cups, only serving with 1 tablespoon. So this is the nutrition facts of my spread. And at last, nut pulses and seed is the source of substances of high biological and nutritional value. There is a need to promote the use of locally available sources of antioxidants because it has a great advantage of include weight gain control, cardiovascular disease prevention, aging control, aging protection, hypertension control, smoothing of the skin and Alzheimer's disease and cancer inhibition. And also contain minerals to encourage beneficial cooking practices through nutrition, communication, education programs in the community. Thank you.
There were about 7 presentations, including myself. We started from the presentation on medicinal plants, natural plants, material which are good for the health in terms of medicine and natural sources. Then I was very happy to see the young professor had a lot of contributions. I was proud particularly when I heard that he happened to be student of Dr. Mir Rajab. Mir Rajab was 1 semester junior to me when I started my MS in Qaid University. So I feel very good. Second presentation was on role of fisheries. Its importance in food production, food security, and its value as vitamin supply. Ms. Kashifa Nagma, she had a nice presentation. Then there was a third presentation on multiple metal resistance in chromium, reduction from untreated tannery. There was an online international speaker. It was probably on pomegranate treatment of leukemia. A lot of pomegranate is sold in Canadian grocery store, and that encouraged me. I will prefer buying pomegranate also is a source for help against cancer. Presentation fat diet was presented here. Dietitians sit in Canada with the health practitioner, and they are paid by the government ORIC. So dietitians, I don't know how much scope they have in Pakistan, but in North America, dietitians are sitting beside the family medicine practitioners.
Present the Shield of Honor to our chair and co-chair and our two guest speakers. Thank you, sir. Dr. Kashfa Nagma. We would also like to ask Professor Jay Perkins to please have your Shield of Honor. Thank you, sir. Professor Dr. Mushtaq Ahmed. Thank you, sir. Thank you so very much.